Please welcome Birte Arlt, Charité Universitätsmedizin, Berlin, Max Delbruck Center for Molecular Medicine in the Helmholtz Association. Pulling the emergency brake: how cancer cells cope with nutrient limitation.
Hello everyone. Every day 822 children are diagnosed with cancer worldwide. And one of the most prominent solid tumors is neuroblastoma. Its aggressive form becomes more resistant to current therapies. So you all have small molecules in your body that are downstream of the DNA and carry information of our genes, proteins, and of our lifestyle. They are called metabolites. So we believe that understanding metabolites is essential to meet the urgent need for new therapeutic drug approaches. We measure metabolites and their corresponding proteins by mass spectrometry-based analysis and integrate the data with different software and programming languages. Following data analysis, clinicians and scientists come together for data discussion. Tumor cells can import nutrients like glucose, serine, or glycine by transporters into the cell. In case there's no serine or glycine available in the surrounding environment, we observed an increase in pathway activity leading to intrinsic serine and glycine production. Moreover, we identified the protein PHGDH as a major protector in neuroblastoma to circumvent nutrient limitation. The PHGDH pathway is converting an intermediate of glucose into the nucleotide bases required for DNA synthesis, essential for cell growth. To test now whether PHGDH is influencing the cell growth behavior, we destroyed the gene encoding for the PHGDH protein by CRISPR. We constructed 5 cell lines that don't express the protein anymore and observed a significant decrease in cell growth compared to the control cell lines still harboring the intact gene. In follow-up experiments, we transplanted a neuroblastoma tumor from a child into mice. PHGDH inhibition showed no convincing effect on cell growth, but chemotherapy treatment stopped tumor growth. In combination with the PHGDH inhibitor, tumor growth was accelerated again. We were quite surprised to see this result after having deeply studied this pathway. So how could it happen? This map is illustrating all known metabolic pathways within human cells, and the one we studied is marked in red. So imagine now you want to travel through a metro system from point A to point B, and the route is blocked due to construction work. You can still travel from point A to point B, but you have to take a detour using another connection. And that is actually true when the PHGDH pathway is inhibited. So only by applying different models and technologies we were able to observe this metabolic remodeling within tumors. I want to thank Professor Angelika Eggert, Dr. Hedwig Dolzer, and Dr. Stefan Kemper for supporting this project, and thank you for your attention.